G'day, good morning, good evening. How are you? I hope you're well. It's going to be a great show today. It's a cannabis show, and I respect the law, and I hope you respect it as well. And today, talking on mutations. Great show today. It's going to be so interesting.
The biggest property boom in history. It actually starts in 2023.
Sorry, that was my tablet. I'll have it on to answer questions so I can keep it up to date. Yeah, plant mutations is going to be fantastic today. All different types of expressions that they put out from shifting genes, or there's different ways for mutations to actually happen. I'll go through heaps of different ways, and talking about polyploids too, having extra sets of chromosomes. Why is that a bonus? And showing different explanations on studies that have been done with polyploids. It's pretty cool. All right. I'll give it a little bit, just 30 seconds. What else can I talk about? Well, I'm just waiting for the live people to link in. That's the string. Actually, I'm going to change the window. There. You know, I'm seeing my blooming little thing going around. This night's working all right. But yeah, some interesting stuff in medical cannabis, some expressions that can be used for benefits. It's pretty cool. Right, I think that's enough. So today, share screen. No one to say good day to. Oh, well, it's all right. I'll come back to that. Well, actually, I should be waiting because there's some pretty cool stuff on. Oh, well, there's 2 studies that have been done in Australia that they want participants for. So I hope people rewatch it anyway. This was on the news. I'll go the studies a little bit, but this is the news first, and then it's just negative. Look at it. So the New York Post put out this ad: weed makes you crazy and gives you heart disease. Why is it legal? See the date, it's recent, a few days ago. It's a couple of weeks ago. And then I found it was ripped in from the Australian News website. So the Australian News website jumped onto this one, and this is, it just goes on a little bit to say that a new peer-reviewed study of 60,000 Canadians were done who had misused pot and have had strokes and heart attacks and other cardiovascular problems. In other words, weed's not as a harmless vice. It's a killer like cigarettes, this ad says. It is so, after all the studies that have been done, actually, what's that? There's a click on that. Peer-reviewed study that I just clicked on. I haven't clicked on this before. No, I doubt it's not peer-reviewed. I'll quick get away from this. Because no one would say that, because there's so many studies have been put out of recent to suggest the opposite. So how they collected the data is at question. It's just to show the way that how medical cannabis is progressing. Some people take the worst things out of it, and they write things that the uneducated read, and they just go, wow, this sounds terrible. It's the devil's poison. You know? Anyway, that's the news. Some better news. The Italian court temporarily blocks the reclassification of CBD as a narcotic. But industry warns it's just the beginning of the long struggle. So over in Italy, they were going to block it because it didn't work, because of probably all the media outlets that are just giving the bad thing. And then they had to produce all these studies, and they found out, it is actually working. That was a few days ago it happened. Next one. Study demonstrates that cannabis is an exit drug, not a gateway drug. So this is a study done a few days ago, I posted a few days ago. Actually, I don't have to really elaborate onto that. I think it says enough. It was a Canadian study. And it's true. It's an exit. It doesn't lead to others. It actually gets you off all of these bad and harmful things that have mega bad side effects, liver, all sorts of problems. Next one, revealed government to announce mega shake-up for New South Wales drug laws. All right, here's some Australian stuff. So I'll just see if anyone's in here. Hello to everybody. Righto, back to it. No one's here yet. Well, there's people here, but no one's really saying much, so that's up to them. So the study Australian bit is the government is to announce major shake-up of the New South Wales drug laws. So a few days ago, they introduced into Parliament a two-strike policy. So if you get caught, it's just going to be virtually like a slap on the wrist for certain amounts. It doesn't say amounts or anything, but that's happening, which is pretty good. There's a few Australian updates here. Australia's cannabis market posts brisk growth in the first half of 2023. Excuse me. Goes down to say that it's just been going very well, as expected. And it wouldn't be, I don't go on to say how the importing compared to Australian grown, because it can't even keep up. The amount that they have to import to the amount that's grown here is just, yeah. By category, prescribers prescribe mostly CBD and then THC, because the doctors don't know anything about, they're not experienced enough to actually go and give THC to patients first up. So they go and offer you oil first up and CBD. Does that work? No. Anyway. Next biggest drop. So I'll just scroll down if you want to read it later. There you go. Next Australian study. Medical cannabis authorised prescribers scheme applications. So this is the applications, the amount that was put in by prescribers to get their application. So you can see over there to license to give it out, medical cannabis in Australia. So you can see in 2018, there was 105, where this year there's nearly 11,000. And over the medicinal cannabis categories, most of them are CBD, which is I'm pretty sure it's legal over the counter. You can get whatever you want over the counter with CBD now. It's the THC above 0.3%. That's the holds all these approvals and money and all sorts of blah blahs. All right, the prescribing consultation locations. Right, so everyone's in Queensland's doing it. In Victoria, it scales down to New South Wales, WA, South Australia. ACT, surprisingly, where it's fully legal. Tasmania, then NT is the lowest. And the dosage form, how are they administering it? Yep, oral liquid. Because the doctors, I can confidently say, they know extremely little and they're not qualified in the herb. So they always offer you liquid first. And the liquid is so diluted, it doesn't even work for some people. You don't get the full spectrum profile in that to get its medicinal properties that work when they enhance it, combine. And I'll just go down capsules, inhalation, wafer and solution, topicals, and then oils down there, sprays and other heaps of different other ways that they can prescribe it. They've made right down to patches, suppositories, tinctures. Tincture should be highest, one of the highest up there. There's only 42. It's one of the purest forms. It's when done properly. Anyway, I'm just reporting what's going on. Up here, Canadian cannabis farms eligible for federal funding via a $3.5 billion for B program. So Canada is just so rad. Props to all the rad Canadians out there. Hello. The government is releasing a $3.5 billion program to fund different sorts of schemes and experiments to realise that this medicine is a medicine. How can we get the best out of it? Good on you, Canadian government. What's this one? How pink? Oh, this is to do with the ornamentals a bit later on. Ornamental medical cannabis. Can you believe you can have such a thing? Right, let's see if there's anyone. Oh, yep. Hello, hello. Hey, off grid. Live. How you going, mate? I'll put that up. Nice to see you. Welcome. Hello, Aaron. Nice to see you. And everyone, cannabis is a gateway to great health for lots of people all around the world. You got that right. Well said, Aaron. Hello, sunshine, rainbows. Nice to see you. So much potential as a medicine, and yet so suppressed. Couldn't have said it any better. So sad. Like last week, I went through 700 different studies to show on how it works in various aspects. But that's not today's show. Today's show is plant mutations, polyploids, ornamentals, variegations. This is going to be a pretty large show. I'll show you all the slides that I've got to go through. And I'm just going to rip right into it. Some cool ones too. Ones you've probably never seen before. Ploidy, no. So this is all today's slides. So there's a few, it's about average. But then later on, I'm going to go through my ploidy folder, and in that is heaps of extra radness of stuff. There's also some of my ploidy slides that I've got that I'll be, oh no, where's the other one? Oh, there's one on gene mutations that I was going to start with. I'll have to find it. Yes, that's what today's shows entails. Let's start. No. Yes. Am I sharing? No, I'm not sharing. Wow. I am sharing. Oh, there it does. Sarah's it up the top. So this is what it's about. Just some weird medical cannabis. What's up with that? I'll just go into a little bit of explanations on what it is a bit later. First, there's a little bit of news and stuff. So Joe Biden, good on him. He's pardoned for the last year cannabis folk that have been charged and jailed even on it. He squashed all of those and got rid of all of these charges. Good work, mate. Keep up the good work. Once the extra world recognise it, especially Australia as a medicinal plant, everyone will be better off. And look at this. I couldn't believe this. This is in Brisbane. This is the Liquor Legends, is a liquor alcohol shop in Australia, and they're selling CBD. I nearly would like to somehow get some just to try it. I don't know if it's got alcohol in it, but it's got CBD in it. How cool. And you can just get it off the shelf. So good on you. Come in and try it, I reckon. Well, that's what they say. That's what I reckon too. See what it's about, if it actually helps you. I found this funny. I showed this last week. I'm not going to show it anymore, but it was in here. Everyone has the right to be happy. They sure bloomin' do, mate. And why can't medical cannabis be legal for everybody to grow? A few days ago yesterday was World Algae Day. Happy World Algae Day. Microbes are rad. This is a pretty cool thing. So you'll probably see this a bit more. Cannabis saves lives. Actually, it should be medical cannabis, because street cannabis actually can harm lives. So yeah, actually, I might have to change that. Thank you for bringing that up. And this is pretty cool. My mistake. I'm not here for you. I thought you were smoking tobacco. Oh. I think that was pretty funny. I hope you're laughing. At me or with me, either one, doesn't matter. And this is it's showing on the slide a brain with a nice leaf, healthy leaf over the top of it. And it's to do with the entourage effect. This was a study done to show that it's not THC that's the main contributor. It's the main indicator of potency is the entourage effect, because it's the contribution of the terpenes, flavonoids that are combined with the cannabinoids. And this is an amazing thing that just shows why you need them and why licensed producers and the big people that produce all the cannabis, it's mandatory for them to install a radiation, big microwaves that they have on site, and they're allowed to do it down to a certain percent, and it passes. This is what happens when it's when they zap it. So many producers expose cannabis to ionizing radiation, especially sterilizing it. However, this process can negatively impact terpenes, the aromatic compounds critical to their therapeutic and their entourage effects. Our medical cannabis takes a different approach. We keep the harvested flower in its natural state, avoiding irradiation completely. Researchers demonstrate irradiating cannabis reduces terpenes by 10 to 40%, fundamentally altering the chemical profile. Patients often reported irradiating cannabis has a diminished aroma, flavour, and moisture. So yeah, a lot of our stuff that you're getting on Australian medical cannabis has probably, I'd say nearly 3/4 of it has been done like this, as sad as it is, because they've got to make their money. So for them to, they can pull it down from 8% with all the mold and fungus growing on it to under 5%. If actually, I don't think it's 5, whatever the rule is, but they can pull it just under it with their machines to make it passable and a sale for them. But this is what it does, mate. It's just putrid. Medical cannabis. This is why it should be legalized to grow at home. Oh, the cool Australian studies. Share this to your friends. There's 2 here, to Australians. Do you use medical cannabis to treat anxiety? If so, they want you for a study. The University of Wollongong is looking for participants to join. I'll give you a gift voucher too, it says, and I don't know what the gift voucher is. I can only imagine. But anyway, it's just good. And so if you'd like to do it, go to the University of Wollongong, Australia website and look up this medical cannabis to treat anxiety and participate. Another study done in Australia where they're looking for participants is, see, it's going to happen in Australia. It's going to happen in about a couple of years. You'll need, they want participants for chronic pain, this study is. So they're recruiting Australia-wide, investigating medicinal cannabis oil for musculoskeletal chronic pain. So if you've got any type of chronic pain, contact these, get on to New Zealand as well. The eligible questionnaire, yeah, I'll just zoom in. Get into that. Go to that /lkd.in/G, big T, small v, big S, big Z, big V, big D, small q. Go to that and sign up, and let's work together to make a difference. It says. But it's good. So Australia is starting, at least they're starting. You won't need all this medical, this rot to say it's not working though. All right, next. Oh, I like this. ADHD, I have a bit of this. So it's trying to do everything at once or you fail today. That's like when I read these studies back to you, I find it really hard just to read because I just want to get to the end of it. So I just want to skip it. So it's hard, but non-ADHD, hey, all's good. So this is what I feel like when I have medical cannabis and not a sativa. When I have my indica or a hybrid up to 58% leaning sativa, I feel like this. I can calm, have tunnel vision focus, everything's peaceful. But without it, some more news. Interesting in carrying cannabis or hemp-derived cannabis drinks in your US state. We'll take a look at this handy map because you don't want to get yourself in trouble. So the green, it's generally an open market, sweet loving, go wherever you want. Yellow, it's more complicated market and there might be restrictions, especially as we go over there on the East Coast. Then the red is just a closed market. It's either so ridiculously one way or it's impractical. It's straight up banned. Red states. Washington. I didn't think it was banned, but anyway, that's what this chart says. So Washington, Idaho, Utah, Michigan, and over here, just banned. You can't have drinks, the hemp-derived or cannabis drinks. I respect the law and hope you do too. This is the old timers. I just like this. I like the old time sort of stuff. It's cool. Old time Dodge trucks. Imagine all the stuff, the oil and the big bales of legal stuff that would have passed around in the day. We were, what's this say? Where we are told the best cannabis genetics are found. Oh, yeah. But where they actually come from. Yeah. They're not, more than likely, they're not coming from a clean laboratory like that. They're from someone who's got a rad farm and they're just doing their experiments and producing the cool stuff. I put this in because I just found it very interesting about a flying fish. I didn't know there was flying fish that can fly up to 600 feet or 200 meters, reaching same as a car around town. It's really cool. Imagine, I just finished a course on biomimicry. Imagine biomimicking this. I'm sure you could do well. And then another way to consume. I found it funny. I think it's more funny, but yeah, it just shows us a straw with a T-piece in it, like a three-way straw, and you can put your joint in it, and just so you can have two things at once. Yeah. Hey, that way would stop it going down your lungs, wouldn't it? Because if you're sucking this, you know how when you open up your lungs, it's going down a different pathway, you cough and stuff if you have water going into it. But this is going straight through the stomach. So this might actually be a medical safe way of smoking a joint. Holy mackerel. Whew. Yes. To the best of our knowledge, THCB. Oh, yeah, martial artist. Hello. I'll go and say good day. I saw martial artist has popped in. Hello, martial artist. Okay, mate. Oh, sunshine's out of there. See you, sunshine. Nice to see you, martial artist. Hello, Blinky. Nice to see you. Hello. Oh, Aaron says, flying fish are really cool to see. Yeah, I would like to see one. Wouldn't want to be sort of hit by one. That wouldn't be very fun. All right, back to sharing. THCP. So what's this THCP, they reckon? They reckon that it's up to 30 times more potent than THC. That's what they find. Can I just summarised it. Real evidence. Yep. So its THCP was found to be potentially 30 times more stronger than just THC. Amazing. That's how the body interprets it. So it's pretty cool. All right, let's today. Mutations. Plant mutations. This is rad. So up here is hauled phyllotaxy, and that's when you get more than 3 nodes coming out of a node. Usually it's 2 or 1. And when you get 3 or more, it's hauled. W-H-O-R-L-E-D. What's this one? B. Look at the leaves. They look like a big fat duckfoot. See at the end different? Hello to Frank if you're listening. Frank plays around with those mutations. There's very good benefits of mutations I'll talk about a bit later. This is some Australian bastard weed in Australia. The reason why we get it is because it gets zapped when it comes in. And then all things happen. They have DNA shifts. I'll go through that a little bit later about how it happens in the plant and the different types of shifts and recombinations you get that produce wrong outcomes for growing like this. And over here, this is actually an ornamental, and believe it or not, it is cannabis. I've got a cool study I'll show you a bit later on ornamentals in cannabis, because now that it's becoming more legal everywhere, people like pretty plants. And why does it just have to be in the philodendrons or, you know, lilies and all those types of things? Why can't it be in medical cannabis? Exactly right. All right, this is to show double fertilization. This is more employedy. I want to come back to that. So I'll talk about mutations for a bit, because mutations are fascinating. What is a gene mutation? I'm going to find it first. Actually, I don't mind showing this. Ploidy. It's close to ploidy. It was in genetics. Mutations? No. Chromosomes. This is it, I think. Halfway through here, the thing that was going to start. I was going to go back to it. It is. Here it is. Right. I think I got it. Ready when you are. No, that's not it. Yes, it is. Excellent. Things are flowing today, people. It's really good. So what's a gene mutation in plants? A gene mutation is a stable, heritable change in genotype caused by an alteration in the nucleotide sequence with the gene of a genome. A gene, genome, cell, or individual carrying a given mutation is termed a mutant. Gene mutations can affect the nature of the gene product or interface with its expression. So all types of cascading effects. It's awesome. I suppose, yeah, spontaneous mutations. Actually, I'll come back to that. You want to see some photos first, I know that. Give us some pictures. All right, I'll come back to this. This one. There's some pictures. So that's what's happened in this plant. So the cannabis plant's got, 35,000 genes, pairs of genes, and each one of them interacts with each other to allow the pathways to give their expressions as we see and as it happens. But when it changes, they chop them out, they cross, all types of codes are different, and it gives these expressions. And that's the leaves, is just cool as. And you can use these to your advantage. I'll go on that later. There's just some different other expressions. So cool. Looks like just a fern or something like that. It just really doesn't look like cannabis, but it would stink radically. I think this one's to do with the foxtail. It's just a big, just growing like it's producing foxtails. Like that's what a foxtail looks like. At the end of your cycle, when it finishes, it'll usually just sit there and it can keep going for nearly double its cycle. But then all of the medicine in here is all fatiguing and dying and changing CBDs, changing THCs, altering. This one, fasciation, is amazing, where it just looks like the whole thing's a bud. So instead of producing calyxes that it normally would, or bracts normally would, it just go and produces massively wide ones and different arrays of them. You can actually, I did an experiment with this, you can produce this yourself by cutting these stigmas in half, or the anthers, cut them in half, and it goes sideways, not up. But that's fascination, Fasciation, is another one. So you're going, that's not blimmin' cannabis, what's he on about? Oh, look at the leaves then, friend, look at the leaves. It sure is. It's full on. It's cool as Fasciation. Here's the old duck's foot. Look at the duck's foot with its feet. It produces not normal leaves, and it's throughout its whole cycle. It's pretty cool. Hello, Frank, who plays around with this mutation. Here's another fascination. There's another cool one that's happened. You can see how the nodes don't, like the leaves grow next to each other too, so it's a pretty telltale sign that it's happening, not just the top. And this one is Whorled, W-H-O-R-L-E-D. So it's where you get more than 2 nodes expressing themselves. You can see here, look, there's like 6. There's actually one fella in Australia who's got this, and anyway, that's what it is. Friend who lives in South Australia, you have a Whorled mutant, W-H-O-R-L-E-D mutant. Same thing here, because it's putting out more than 2 nodes out of it. So that's nothing to do with a triploid or anything like that. It's called a Whorled mutant because it's activated that pathway. Polyploid cannabis. Polyploid is a mutation in nature in which cells get an extra set of chromosomes. So this is not, these pictures are not correct. So I just pulled this slide out because I thought, well, that looks rad, but it's not actually correct because these all have still the same sets of chromosomes. Polyploid is only to do with chromosome shifts, and this is a mutant shift in the genes. So this is where it's shifting chromosomes, this is where it's shifting genes. It's the difference. These are some funky, oh, Main Green, how you going, mate? I'll say good day to Main Green. Hello, Main Green. Hello, cheers. Hope you're doing well. Today's going to be a rad one. I've got some fantastic studies and things to show about the mutants and polyploids later. So I'm talking about mutants now, and then later on I'm going to go on about polyploids, why they're good, etc., etc. So I'm still going to go off the slides here, and then I'll go down to that little bit. So this is, I liked these trichome pics, and I thought, wow, that's pretty funky looking. You know, they must have stained that, have they? Nope. What this person's done, I think they've ruined it personally. They've gone and just coloured in the bits, made them look cool. That's an electron scanning microscope, and that's not how it should look. And I think It might be art, but I personally would rather just be looking. I want to see these sessiles. Look, you can even see the description in the side of the cell, in the head. Sorry, it's awesome. So bad art, bad cannabis art. Just wanted to show that. That's personally what I think, though. Maybe other people think, "Wow, that's so cool," but it's not true. They've genetic. That's not. They've coloured it in. So it's called fasciation. It's not a polyploid. This says, often confused with ploidy. Fasciation is an abnormal growth pattern in the flowers or stems, the stems of a plant. It may be due to a localized mutation, induced, spontaneous, all different types of mutations, pests, anything that cause mutations. These, I'll go on a little bit later and show you how the different mutations can happen briefly, actually, because it's, gosh, Mendel's genetics is full on. These plants have their normal set of chromosomes, all of them. All these plants have 2 sets, yes. So see in the middle, when you see the stems that are squashed, that genetic expression is called fasciation as well. So it's not just in the heads, in the buds, it's also in the stems. So you see it's sort of flat and skinny, and I've seen that a few times, and I haven't been able to put my finger on it. Now I will be able to. Fasciation. This is more ploidy stuff. Here's the size of the polyploid seeds. Tetraploid seeds, sorry, they got 4 sets of chromosomes in them, and these only got 2. And they're extra small too, some death bubble 1-to-1. But yeah, that's a massive 1/4 of an inch seed. Hey. Oh, looks like we're into the ploidy section now. Oh, very good. That main green AI trichomes. Yes, that's exactly right. This is some cool stuff, some different types of polyploids. Now, I'm interested, anyway, I'll just go on. Autopolyploids are when you have one genetic set and then you'll double it by a chemical, usually a chemical process. The allos are the rad ones, and that maintains the hybrid vigor, and you get your fast and vigorous growth. And that's because there's 2 parents that's introduced into creating that polyploid. So you're still getting that cross, that genetic difference. With this one, you're not. When you do inbreeding, you can lose a little bit of the DNA code because it chops out a little bit of its code because it might, some of the sequences mightn't be correct. So that's what happens with that. This is a mixture of sort of both of them. Best way to describe the segmental and auto allos. Hey, happy man. Nice to see you. I like that. Brings a smile to my face. I hope you got a smile as well. Oh, and this is some thank you. Thank you for the support, for the membership. So $5 for support. Thank you. I appreciate that. The next one, chapter down, is $80. It gets you my private playlists, about probably 15 of them. $150 gets a 20-minute video call or a novelty gift like a chart or something that I made, and $300 gets a weekly video call, and that's more so catered for people who are going through doctor's problems and want my educated opinion, or for commercial growers who want to start up legally and do the right thing. Canna jobs, entrepreneurship in cannabis. How can we do this? Hey, Frank. I was just giving you some props earlier, Frank, about some duckfoot. You missed out, mate. Actually, let me know if you want to come up and have a chat. You can describe your duck feet. I'll give you a link a bit later after I go through this. Entrepreneurships in cannabis. How can you cool folk make money out of it? Well, if you're artistically inclined, you can put this fella in, I think that's Jimi Hendrix, into here and sell it. Or probably not sell it because he'd probably copyright it. But anyway, this is just an idea of what you can do. Have fun. And this is for brewers. This could be infused wine. This is the first THC infused wine. Wow, well done. And this is for like Kenna Cribs or something like that, where you've got your Airbnbs and you want to cater for all types of folk. So it's nearly legal everywhere now. So yeah, get unique in your market. And this is some more drinks that have been put out. The drinks, there's so many of them. We don't see, well, we've seen one in Australia I showed today. Props to that. But yeah, so you brewers. What's this one? Hot off the presses. A study looking at mutations in cannabis tissue culture. Oh yeah, this one showed that before anyone starts freaking out about tissue culture, we have previously found mutations within a single cannabis plant. So regardless of propagation method, cutting or tissue culture, mutations are a reality. So there you go. So it depends on what your genetics is like. Mutants can happen even in stable tissue culture scenarios. That's what this study says. That's the summary of it. Ploidy and mutations. Here we go. Look, there's a bit of duck's feet down here. Do you recognise this, Frank? Oh no, it's not on this page, sorry. Polyploidism, freak show, and mutant cannabis varieties. Polyploidism, well, that's about the chromosomes and the fasciations. Went about that before, about that gene expression from the stems or in the flower on the top. And the freak show, was a mutant. This is pretty cool. This is what Frank does as well. So if you should work with these weird mutant varieties, because you can get something that's going to be revolutionary down the track. In 30 and 50 years, people won't be talking about medical cannabis. They'll be talking, oh, what style of mutant or whatever you have. Do you have the duck's foot from Frank, or do you have, something else? So this is one from Jordan. So he's named it Freak Show. So it's a fern-like leaves, like the ones we were looking at before. Should be called Freak Show then. Okay, just for those who missed it. Oh, doesn't even have to go back to this. Oh, that's what this little thing down the bottom does. No, it doesn't. Anyway, there was those fern-like leaves at the start. So that's called Freak Show. All right, I'll have to remember that. That's what he's named it. And he won an Emerald Cup with it. So now you could be crossing that and getting that expression and working with it to your advantage. So it's a call for experimentation, embracing the unconventional. As the industry verges to a new, from being medically recognized in the planet, it's going to be recognized everywhere. So that's why we're going to be variegations and all sorts of different things that are coming out of medical cannabis. So this is a new saga that's starting to happen. So get with it, do some stuff too. You'll be a leader. The existence of polyploidism in the event of freak show, well, it's not a polyploid, it's just a gene expression. By nurturing a vibrant and flexible gene pool, we can sidestep the risk of confining ourselves to a corner. Oh, okay, I'm not going to read the rest. What's this one? Ploidy. Does the protein content increase linearly with the number of chromosome sets? Okay, well, no. No, it does though. Oh, here you go. As you get up here, it just shows that it increases a little bit and can taper off as it gets up to higher amounts. I'm not going to go into that one. This is some terpene, some trichome stuff. You can see this is the Trichome Institute, but I just like the picture. You can see, look at the unitrichs, stalk capitates, and you can see the cells inside of them. That's the bit I like. Dermal tissue, all the cells on top. Very funky. These are some natural trichs. There's no AI trichs here, like Main Green said before. These are some cool natural ones. You can see they're not pink or purple because these aren't reflecting the same. So that's just a natural gene expression. And I'd have to think a lot of hemp cultivars express this, so it might be hemp. Not quite sure. This is some trichomes then that want to be harvested in my eyes, because it's just a good amount of ambers. Any more later and it's going to really deteriorate. So you can see in here the three difference. There's the clears, see-through, milkies are there, and ambers are like that. So that's a good depiction. And you shouldn't be looking at the leaves. This one is on the bract or in the calyx. So it's a good way of bud maturation. Not plant maturation, but bud maturation, because the leaves will show a different sign way earlier. And then the bracts or the calyxes show next. And then the overall plant, you'll see it just starting to die off, and that's a plant mature. Here's another rad thing about the trichs. So they've gone and separated this using what's the technique? Our stable solution can separate particles based on their chemistry, allowing us to isolate glandular heads from immature heads. So they can get this tiny, tiny amount of green leaf material and even the immature heads to have a mature head outcome. So that's pretty cool. So that's going to be more stable, probably like all of the cultivar will express its full spectrum, and you'll expect to see it very evenly throughout this mixture rather than the immature ones. So that's pretty cool. Has there been any questions? Oh, Main Green said some stuff. Sorry, Main Green. Ploidy, yep. Everything's legal in the same states. Tissue cloning, Brixol, yep. He was in the hangout that us way back, bro. Oh, if it's the same dude. Yeah, it was Frank. Yeah, Frank and Beans. Yep. As his cultivars stuff. This is just nice. I had to put in because it's just blooming nice. It's got nice senescence. Look, the anthocyanins are showing. Very medicinally healthy. Chunk. Actually, looks a little bit like Pink DB. Done that, Main Green. This is nice too. Actually, looking at it, it's not all that sort of nice. There's a bit of a health problem down there. It's, in general, it's pretty good. Oh, look at this different cultivar. Ugh, look at the skinny leaves. Ooh. Actually, there's 3 different there. Wow. Oh, I see. Look up the top. Right. It's got some fat leaves too. It's pretty interesting. Wonder if, yeah, that's some good work. No morphogenic mutations like the others in here. That's just nice, isn't it? Wow, that's the end of the slides. So now I'm going to go back and show about the mutations, spontaneous mutations. So Frank, no, that's cool. Spontaneous mutations are generated by lesions and rough handling of plants. So you can go and injure them and that'll induce a mutation. I'm going to really skim through this because there's so much different things to go through here. Induce mutations, when you induce them. So that's what Australian bastard weed's from. So that's because it's induced through a mutagen and the agent is radiation, where you can do it through all different types of chemicals and ways through here. I'm not going to go into. Here's some physical ways. That was the radiations, UV also, and rough damage is another physical one. Biological, enzymes. What's this? Mutations, random or adaptive? They're both. They're random and adaptive. So this is just a breeding chart. So you can see the expressions from breeding from induced mutations and the expressions you're going to get through 4 progenies of spontaneous mutations. Mutations based on their location. So there are somatic mutations, which is those occurring in any cell except the germ cells. Somatic, that's what this says, germination. I can't really read that. It's a bit blurry. Those, no, not going to read it. Mutations based on type, point mutations. Okay. And there's nonsense mutations, silent mutations, missense mutations, transition mutations, frame shift mutations. This is how the frame shift one works. So normally the DNA code should be like that. That's what it expresses at the top. It's not written like that because it is 3 letters, but it's the N-A. No. Oh gosh, this is testing. I haven't played around. Oh, it's G, guanine, adenine, thymine, GAT, and C. And that's in the, anyway, look, and the frame shift mutation is when one letter just changes, it's substituted in here for a point mutation. For a frame shift is when the whole thing shifts 1, so this whole code shifts 1 to the right, for instance. So and this one's lost the C, so it's shifted to the right and it's just lost. See how it's all different, weirded. This is frame shift, but it has an insertion in there. See why I'm just really, really mega touching on this? Because it's so much detail. Mutations based on phenotypes. So you get more mutations from a heterozygotic plant that's not stabilized. So if it's stabilized, the genes, it's homozygotic, and that's why land races don't have many mutations because they're more stabilized. So that depends on the actual phenotype and the genotype as well. The phenotype equals the genotype plus your environment. Applications in ploidy. Polyploids, have I finished with mutations? Yeah, there's no real questions on it. So what I see, actually, I better stop this because sometimes my tablet's weird. No, there you go, I'm up to date. Okay, share again. What's this up top here? Right, to the study later on. So I have to explain what ploidy is first. Applications in ploidy. Well, ploidy is the chromosomes, explained it. So ploidy has been a major force in evolution and speciation in land plants. 50% of land plants, mostly land plants, are polyploids. General polyploids are more tolerant to drought and cold, as well as a higher resistance to pathogens. Actually, tobacco has 4 sets of chromosomes, and tobacco mosaic virus is resistant to it from increasing the sets of chromosomes. Polyploid has been used to produce superior crops like triploids, seedless, banana, and watermelon. Yeah, triploids there, 80–90% seedless. Fantastic for folks. Yeah, go on about triploids later on. This is polyploids. Oh, well, how's the category? First, you got your diploids. Is it sharp? No. I'll just read this one. Polyploids. Why are they all so good? They produce larger fruit, like triploids. The chromosome doubling helps to overcome interploidy barriers. So it's more interploidy barriers. So it's more stable, I suppose you could say, because it's got an extra set of chromosomes that aren't doing things for the plant, so it can act those genes whenever it wants. Polyploids to enhance the production of metabolites of commercial substances. Yes, so they'll actually give higher secondary metabolites. The sesquiterpene terpenes has been shown to be higher in secondary metabolite profile of medical cannabis. One way to identify how to do it, it's a squash method under the microscope. So you can go and do this process and identify your chromosomes under your plant tissue and see if you've induced or induction was successful. Triploids. Let's go about triploids. Triploids, these are the ones that are 80% to 90% sterile. That's why they don't produce seeds. Each cell has 3 complete sets of chromosomes, where polyploids has 4. Hexaploids has 6. Octoploids has 8, like strawberries. And triploids have several desirable traits, like larger leaves, fruit. They photosynthesize faster. The flowers are bigger, greater vigor, higher biomass, so higher yield, and a higher stress resistance. Great, a lot more economic value. Fertilize, that's how they happen. So you've got your normal cell that through meiosis it'll halve to its haploid cell, then they'll join to produce a diploid cell. That's your 2N, but when it happens for 3, you've got your 4 at the start, which halves to make 2. So you plus your 2 with the 1, and that's how you get your 3. Or you could have an unreduced meiosis, and that's how it happens. Another way that it happens, and you mix it with your normal, and it comes out to be a 3. I'm not going to go on about this. There's quite a few more to go on about here. There's a question. I'll pause for the question. I saw it there, Aaron. Aaron asks, why do mutations seem to only appear on one branch or part of a plant? How can I explain that? Because it's when the mutations in the could be in the cell, the genes put out that expression, and it could appear first in that part of the plant, and then they happen when the cells are seed meiosis. This is when it's dividing. So when the cells are dividing, that's when there is a shift in there. They don't divide regularly. I did a course called Cell Biology, which was a course just on cells, and when they go through their different phases, they had different things happen, and that particular one in that part of the plant didn't happen correctly. So it shifted incorrectly, and you got a different gene expression when it's doubled. That could be a mutant reason. There could have been, like I said before, from induced mutation. When you just knock it, you just give it a knock. That's when you induced it, and that's damaged the plant, and it's formed a different process to give a different outcome. Because remember that it says before about the, oh, it's not on this chart, about the depends what environment you're into, because that makes a big difference. Body mutations seem to only occur on one branch. And then if it's a full-on mutation, it could be systemic, and then it'll go and give the following breeding, the following plant growth development after that point will be of that mutation, because that's when it will have shifted its genetic code, because being a mutant. Did I explain? Let me know if I didn't explain it. I can help you a bit better. Sorry, mate. I hope that helped. Methods of triploid production. Yep, that's the chemical way of doing it. This one here is very dangerous. I don't suggest try it. If you're going to try it, use ones like oryzalin. Not that one there, because that's very, very lethal, very dangerous. Methods of triploid production. Oh yeah, here's how it happens. Like that chart I was going to show before. Good time to bring that up. It's a lot better than this one, I would hope. Let's have a go. Back at the start. There it is. So when the pollen grain, it lands on the female bits. So this is the pistil or the stigma up the top and styles down the bottom. So this is, and it's the calyx, the perigonal bract. And so when the pollen grain lands on here, it'll start and put out this pollen tube and germinate. If it's compatible, it will continue. If it's incompatible, it'll stop. So if it's compatible, it'll continue all the way around. And ones that produce double haploids, or they're going to make more sets of chromosomes, they don't go into this egg cell, because the first one, sperm cell, it's got a calcium channel at the front leading it, and it goes into the egg cell, and it'll start and form the diploid in there. So it's 1N and 1N equals 2N. But in the center here, the central cells, centriole cells, they've got 2N already. So if it makes it past it and into here, see how it's 2 sperm cells in here? One goes in here, fertilize it, well, there's one spicking around. If it doesn't go next door to it, it can make it into here, which that's another way where nature can produce a triploid. Cool. There's another polyploid thing in here too. Larger seeds is another example of polyploids. No. Oh, well, I'm sure there was. No, back to this one. Polyploids. Oh, yeah, that's a better one. Application of the triploids. Seedless. Yes, the seedless varieties. Well, more so for cannabis, because medical cannabis, because if you've got a triploid, you're going to get the polyploid traits, but you're going to get the sterility, meaning that it's been very hard to herm out. So it can herm out because they're not 100% infertile, but try to do it. I worked with one for 2 years, induced, tried to do everything. And at the end of the 2 years, I had to, well, right when it went through hard stress is when it produced only a few pollen, I didn't get pollen grains. No, I was able to pollinate it. So it was successful, and that's where it produced the polyploids. Applications, prolonged flowering. So flowers are generally larger, more colourful and desirable in triploids. Flowers have longer shelf life, which is so special. That is so much needed. Our Australian medical cannabis, it sits in quarantine for a few months. Bang, there goes all the terpene flavonoid profile. It's not medicinal anymore. Aaron says, oh, that was good. Oh, good on you. Thanks, Aaron. Applications of tryptoids, yes, they're more photosynthetic. So they're larger somatic cells and guard cells, the higher number of chloroplasts, so they get better photosynthetic activity, which gives you vigorous growth. I've got a good photo of that. Actually, where is that photo? I was going to go in for more in too. Where's my photo on mutations? No, ploidy. Ploidy. It's in plant science. No, is it? You've got, because you're all watching now. I don't know what I'm looking at. Plant science. I'm in here. Ploidy. Oh, it's in here. Ploidy, there it is. Oh, okay. Probably a good time to, I'll discuss this now because this has the, I should have said this at the start, the terminology. Sorry, this is the diploids, 2 sets, monoploid, 1. Haploid is when it halves their breeding amount, so it starts off as a diploid, it'll halve and become a haploid. It starts off as a tetraploid, which is 4. If it halves, it'll become a haploid into 2. Polyploid is more than 2 sets of chromosomes. That'll do. And I think I've been over the last few. Oh, euploidy, I haven't. Euploidy is possessing full sets of chromosomes, having extra or missing chromosomes. So it's when you've got 1 set more, 1 set possessing full. No, it's just having extra or missing. Yeah, it's the, I'm not sure about. It's having extra or missing. It's 1 higher or 1 lower, like a chromosome imbalance. Lopoploidy, hypoploidy, under or over. Pseudodiploidy, false. All different ways. Induced mutations, I went through them, good. Applications. That's where I was up to, actually. Sweet. Oh, I was going to show that. I remember I was going to show the sets. That's examples like this. That's in my other folder. I know where it is now. This is where it's the difference in the plant. So see this one on here. This is a tetraploid. So this has 4 sets of chromosomes. This has 2 sets of chromosomes. So you can see it has a larger middle leaf. That's a definite telltale sign giveaway. The larger middle leaf looks just full on. It'll look abnormal in the whole of the growth. And it's only on, it has to be a mature leaf, not one that's growing or maturing. Because all these tests are done on mature leaves, the size of the stomatas, the stomata density, all those types of things you can see in the morphogenic expression. They've got thicker hypocotyls, the stem's a lot thicker. Let me show you that. Let me show you those. I got it. Where's ploidy? There it is. This one. This one. So this is a field showing the difference. So you can see on the right, this is a triploid, and the left is a diploid of the same cultivar. And you can see, well, look at the difference. It's, look at that middle leaf there. That middle leaf is as wide as my hand, that leaf, like pretty much the hand on here. And over here, it's just normal. A lot better expression. There's a scale on the left, scale bar. Down the bottom is actually when it's been harvested. You can see the difference. So on the right here, look how full it is. Just a lot more growth from the size of the photosynthesis production from the massed leaves. That's normal, what you would say, or was normal. Next. So the production of trichomes is down here with the tetraploid, and the diploid's up the top. So you can see on the bottom here's a little bit more. Personally, I haven't found this, but this is what this study shows. Here's a stomatal density test. So for the, and then you say, how do you do these tests? Under the microscope. I'll show you a bit later then. So you want a stomatal density. This is just one study showing their stomatal length. So see how the ploidy ones are a lot bigger. So it's very obvious for morphogenic expression and a lot wider. Density is higher too with the diploids and a lot lower with tetraploids because they're a lot bigger, so they don't need as many. That one. There you go. See the middle leaf? These are all uniform. The middle leaf is just, compared against, it's just abnormal. It's just a bit bigger. Same with the stomata down the bottom. Look at that stoma there. That's humongous compared to these ones on the side, and that's both the scale the same size. No. Comparing morphogenical traits of diploid on the left to tetraploid on the right. You can see down here underneath as well. The unicells, intrichomes. What's this one saying? Induction, no. Seedling height. Okay, that was, yeah, it's not very conclusive. This it definitely is. You can see on the right, this is tetraploid in the middle, triploid in the middle, and tetraploid here. So it's 2 sets, 3 sets, 4 sets of chromosomes. So it's a massive, under the same scale. See the scale bar? So the polyploids, tetraploids, more so, have a lot bigger. So you put it under the microscope and measure them. That's what I did. Saw that one. Cytometer is really accurate way of doing it for a tool online. Like Lars Larson knows. There you go, Lars. Just finished replicating my horizontal treatment. Well done, mate. Lars plays around with this stuff. It's cool as. Germination growth of inductions. These are the flower size. You can see diploid to a diploid small, tetraploid bigger. See how it happens. We spoke about that. Here's exactly the same. This is pineapple pink death boa, and you can see here is a tetraploid, and this is a diploid. Look, just at exactly the same age. The seed induction treatment was done at the same time this was at a seed. So it's, look at this really, you tell me the difference. Look at the center of the leaf. It's humongi compared to the other ones. And that stem is so thick compared to this. Same age as well. You can see this pot is sitting on top of this pot. And it doesn't even get to the height. This one's praying, wanting everything for its maximum photosynthesis. No, it's struggling. That'll do, I think. Oh, yeah, there's some seeds. The seeds are always bigger. Leaf size. This is a test done to see what type of calcium, potassium, magnesium, phosphorus, sulfur levels were in the ploidy levels. So you can see how they're a little bit higher, what they absorb. That's pretty cool, but you need a test kit to do those. So it's not easy for the average person. The effects of ploidy level on soluble sugars, reducing sugars and protein in leaves and roots in cannabis. So with the diploid compared to the polyploid with 4s, it's a massive difference. You can see it from its holding ratios, because you know the sink and source cells, if you've got a lot, if the polyploids have bigger sink cells, they're going to be able to absorb more and be able to get it from their sources, get it to their sources. Effects of the ploidy levels on photosynthesis. So there's a lot higher amounts of photosynthesis happening in ploidy level. This is how to measure your stomata. So see the little stoma in the middle, and what's this one using? Within one view. So yeah, you have to get your view to equal 1 inch. That's the key. Then you can do your maths. So you can do a square density per square inch is what you do. Well, that's how I did it. You can see the difference here. Oh, that's induction. That's inducing them. So that's the telltale sign it's induced. See that? So Lars would have seen that. The hypercuddle looking very, very thick and short. It's a telltale sign it's had its chromosomes been doubled. So you're happy. Oh, I'm happy when I see that, because that means my induction treatment has worked. You can't do it forever too, as Lars knows, because, yeah, is that it? Sure. Anyway, no, there was some more stuff in Ploidy. I can show you my stomata test. So this is, you want to put it under the microscope and pull out your scale. And if you've got a good compound microscope, you have to calibrate it first to get the scale up, and then you'll be able to go and pull up the stomas and measure them. So where's a better example? There's a tetra. Here we go. Oh, there's a triploid. So you can see just from here, I've gone and measured the widths and the lengths of the stomas, and then I plot them down on a graph, and then I can see what's going on. So you've got to do the size per square inch, and then you'll know what the density is. And then you can compare that with what the other folk have got, and it should match. You know, it should slowly go up because they're bigger. That's all I really want to share on that. Because I've been working this is my work over the last few years. I don't really want to show too much. The seed test, okay, good. Cotton meristem, okay, very good. I think I've covered that. What else is there to talk about today? Is there any questions? Oh, Lars has been saying, I'll take it to Lars anyway. We can have the screen on the background, which is better. Hello, Lars. Oh, Mace, no way. Yep, there's Main Green. G'day, Main Green. Aaron's question. Thank you for that, Aaron. Your good feedback. Lars just finished playing around with the treatment of doubling chromosomes. Awesome. Hope it worked, mate. You see your little thick, shortened hypocotyls with a bit of red stress into them. Mace, brother, how to see you, mate. It's good to see you. Lars says he's going to use. Okay. You don't have to. There's another method for using stomata, for viewing stomata, where they put nail polish onto it, and it freezes them into their state, so they stay locked or closed or open. I don't worry about that. So I just cut it and put it pretty much straight under the microscope first. I don't worry about sealing its state that it is at. I hope that helped, Lars. YouTube finally notified me. Oh, rad, about time. Yeah, I've been hearing a lot of people don't get notified for my shows because it's medical cannabis, and that's sort of squashed everywhere, even though I've been recognised by YouTube because I'm a cannabis scientist. I contribute to the medical cannabis industry, and I promote the law and respect the law. What's Lars said? Yeah, you can tell they're polyploid when they're seedling, but if you're using clones, it's hard to tell. Oh, yes. I agree. Yes, I haven't done any really clone work with it. Hello, Lars says. Lars, yeah, you don't need it. You can just put it straight on. Very good, mate. Righto. What was the last bit? Chromosome mutations. There's some cool stuff I'm going to show in this. This is pretty full on. Just to finish off. I'll show you those for those latecomers too. I'll just show you I'll show you right. I'll go through. I'll finish the show first and I'll show you all the rad pictures at the start. So the chromosome mutations, I was going to go. Hold on. Where are you? Mutations? This one. Big. So what happens, it does happen in plants, humans, anything, in chromosomes, because that's how genetics works. And see here, this is in humans. Clubfoot, no, can't remember. Chromosome, went through that. That's the definitions, the numerologically. Polyploids is what I should have read at the start. It just shows a bit more in depth about it, how it can happen in plants, animals, and it affects cell sizes. That'll do. Auto and allose, I went through that, saying how the autos is by itself when you induce, and allose when you have two different types, which you get your hybrid vigor expressions. I found here I don't get vigors, but here I do. If I do an induction with this, I don't get, it just grows in like a blah. For the first little bit, it's sort of a bit slower growth, but through an alloploidy, it's actually faster, and it's actually, that's the one I showed before. How ploidy arises, no, done that a bit. Origins, examples of aneuploidy. endoploidy. I went through those before. Hyperploidy, pseudoploidy. Yes, in humans, aneuploidy, look, causes this. Count them. You can count them. Unbelieve. Yes. So you don't want chromosome shifts in humans. Happens all sorts of syndromes. Not very good. But in plants, we can work it to our advantage. Great points. Okay. This goes on a fair bit about some detail. Drosophillas, they're the flies. They've done some good studies on the flies as well, just to find different expressions that happen. And you can compare it because breeding is very similar across the board. It's to do with cells, when cells divide and when they recombinate from when they come together. Structural chromosome mutations, I don't know. Balanced structural, yeah. Skimming through this to show that all the different various types, I could stop on this and elaborate, but there's no point because it would pretty much bore a lot of people. But it's very involved. Or it can be made to be very difficult, but I'm trying to skim through it and make it legible, I suppose. There we go, and I went up to the gene mutation where I read off before, spontaneous and all the different types. That's sweet, that's it. Now back, I'm going to show you some, we'll go to some rad mutation photos that you missed out on the start, all those looking ones. Frank's duckfoot. So this is hauled, where the, I'll show a bit better later on. This is the duckfoot here, where it looks like a duckfoot. There's only one little leaf that comes out, big mane looking fat leaf, see down here, or three, looks like a duckfoot. Frank and Beans is his show, who focuses on that mutation. This is Australian bastard weed, which is, it's everywhere. We've got all different types of Australian bastard weed because you've now seen from the different types of inductions that can happen, and one main one is radiation. So when it comes across the border, zap, it gets zapped, and then it shifts the genetic DNA. So things come out differently. This is variegated. Oh, I got a variegated study to go over. Where's that? Actually, I'll do that right now because I haven't really finished yet. Oh, there's a few up here. Oh, far out. How pink is too pink? Sort of for variegations, I thought. But no, this was to do with, everyone likes to add a bit of colour, but this is to do with anthocyanins. And they were saying there was just a study to do a different pathway to produce anthocyanins through drought mechanisms, during drought. I've done a good study on it and found that it's through temperature. And you can see that on one of my videos, unlock your anthocyanins or bring out your colour potential. And that's especially good for extractors because there's so many different anti-benefits properties that are in it. Oh, man. This is one of the best books. Lars, take note of this, mate. And say you can thank me later on. Look at this book and get it. That's all I'm going to say. It's called Polyploid, Recent Trends and Future Perspectives. Published through Springer by Tanvir ul Hasan Dar and Riaz ul Ranman. I have to give him props because this is one of the best books, mate. In India. My studies are in India. I just finished my university, actually, for this semester. It was excellent. So I'm going to touch on this. This is Ploidy. This is one of the best books that I've read for a while. Introduction. Polyploids. Yes, refer to more than the two genomes. Yes, I'm going to skim over this. That was that chart that I got at the start out of here. Historically, they happen all through time. They happen actually through hardy events. So Mother Nature will be going through drought and it'll just really, the plant will nearly die, and it'll just shift one cell. Bang, it'll double its chromosomes by doing it, and then it'll rejuvenate. So a lot of hardy cultivars, or actually genes of plants, they've, to get through environmental problems, they've done this naturally in nature. So that's why I said earlier, 50% of the plants are ploidy. Strawberries has 8. So that'll do for that. I'll summarize that bit by telling you that. Yeah, for his discovery of a few drugs that can do it. All right. Righto. There's a technical way to work out the base number and then the chromosome number to work out the actual number of the plant. You're going, what the hell, man? I don't want to hear all that. We're not in class. We just want to know a bit of stuff about stuff. I'm hearing you. I'm hearing you. I won't elaborate. Polyploid complex. And the base numbers, yeah. Well, there you go. Read that if you want to understand it. Pause it right now. There you go. Look down. Yeah, see, the highest one ever was 96 ploid. Isn't that full on? Because you times your base number by your actual number. Pretty cool. Aneuploidy. Aneuploidy refers to a condition of having fewer or more sets. Remember before that chart said it was a bit irregular? That wasn't, I wouldn't believe that to be true. So this is what I believe to be true. The aneuploidy has more or fewer than the exact. That's one study. Yes. Next one. History. This is what I want to show. Polyploidism and diploidism. This is a little cool chart to show its process. There's many ways that can happen in nature and through chemicals, because all it is working with cell biology. So this just shows numerous different ways that it can happen. They interact with each other and form different outcomes. So it's diploids all up here, then this is a polyploid, and then it can actually revert back, and then you get your diploids. There's another way it can happen, hybridization. Autos, no, that's the stuff that, that you induce them, and that's by doubling sets, but I really like allos. That's when you have two sets of parents. That's when you're getting your hybrid vigor. So there's been multiple tests and studies that have been done on auto tetraploids, but I haven't seen one from allo tetraploids. And that's what I've said in all of on my LinkedIn things that come up. They all do these studies. They all produce this. They're saying it's less vigorous, less this. That's true because you're inbreeding virtually. So wait until, I keep saying, wait till the studies of the allos come out. It'll be fantastic. There's something else that's cool down here. I want to artificial induction. That's the different ways that we went through before that it can happen. Radiation, temperature, injury is a bad one. Unreduced pollen. Nitrous oxide, can you believe, is another induction way. And there's chemical ways too. That's just showing some difference on the right to the left. So you can see the massive induction, the massive polyploid response compared to diploid. Methods of induction, okay. I think that'll about do of this one. Yeah, we're talking about medical cannabis. Just in case, I'll skim down because it's a fascinating book. Oh, that's how to do it. Okay, that'll do for that one. This one says, new insights into ornamental applications of cannabis. As it's becoming legally accepted throughout the planet, more people are going to have it as an ornament soon, and they want it to look like this. Why can't we have variegated? Why is it always philodendrons or ferns and stuff? Let's have some cannabis. I agree with you. Why not? So this is a study that goes on to say it can happen. And they've looked at it, and the characteristic growth habit, green foliage, aromatic influence of cannabis that provide our ideal profile for ornamental plants. However, due to the legal barrier, the horticulture industry has yet to consider this ornamental relevance of cannabis. So as it's slowly unlocking, it'll become another area where it can be focused on. Just rad. Back at the start, there's a few studies for those people that come late. Australia, big profits first half of the year, unbelievable. Australia has revealed the government to announce a major shake-up of New South Wales drug laws, where they're making it a bit of slap on the wrist, two-strike policy. So it's pretty cool. I don't know what amounts though, so you'll have to look into that. Make sure you respect the law. This is a prescribers to show that through the Australian prescriber scheme in Australia on how many applications have been done and what areas they've done. Massive amount of applications. Queensland takes precedence over Victoria and New South Wales, and their cannabinoids. And of course, doctors always say, oh, liquid, just liquid, because they don't know anything about herbs and the entourage effect. Pathetic. This is also, Australia has this website, this one here, and I pulled off this snippet from the New York Post. Now they said that weed makes you crazy and gives you heart disease. Why is it legal? I couldn't believe it. I saw that. I thought, this must be a bloody joke. They reckon it's a new peer-reviewed study. What a load of shit. So it's, yeah, anyway. Look at my last week's show. 700 studies into medical cannabis on how it's effective, and how it works with cancer and every single disease and problem that there is just about. If you get medical cannabis and not street cannabis, that is a massive difference. In the studies, there's 2 studies in Australia that folks should know about. Also tell your mates, because we need to get other cool people involved. Where's that? Slides. This one. These 2 here. So if you have anxiety, there is a study here from the University of Wollongong that wants you. So do you use medical cannabis to treat anxiety? They want you. So get onto the University of Wollongong and see how you go. You've got to be between the ages of 18 and 50 and be currently using medical cannabis. And they give you a gift voucher, whatever that is. Here's another study. They want to recruit people Australia-wide that have musculoskeletal chronic pain. So they're probably going to supply you with oil. Oh, participants will be required to complete that. Well, costs. Yeah, so they're going to supply you stuff, and you got to do it under their thing. So it's just another suggestion for people. Not to pass the good word. Oh, and let's finish off with some rad pics. This is on my thing, isn't it? Yeah. Where's these rad pics? This is all the mutants I wanted to talk about. There's any mad show. Thanks, mate. Oh, yeah. What's the last say? Here's a hunk of... Oh. Oh, yeah. Okay. All right. Ship some parcels around, mate. Get some radiation. Go through some airports. Let's see some photos. So I went through that. That's the pollination. So this is the called... Oh, what was it called? Freak show. This is a freak show mutant because of the leaves. Very rad. There's freak show. Freak show cannabis, isn't it? It is a freak show, but that's what the designer has called it. So we're going to stick with that. So this is just foxtail mutant. You just got only foxtailing. That's a poor yield, and I would not probably work with that characteristic. It just doesn't, unless it's going to give you a good result. Fasciation is this expression in the flower tops and the stems. That's weird. That's not cannabis, mate. You're tripping, aren't you? Oh, look, oh my, we need this mutant. We sure do. Yep, you can work with all these mutants. They're rad. Like Frank's worked with his Frankenbeans. Look him up. Duckfoot, nice fella too. So he mightn't be getting the characteristic he wants, but he's getting the expression out of the leaves, and it could be an ornament down the track. And then cross Duckfoot with another one, so you'll have Duckfoot leaves, but different flower tops. So this is why you can work with mutants. Like, look at Freak Show, how rad that is. That looks like nearly a pine plant sitting in the bush. It's just something different and unique. So you can cross that with Duckfoot, or you probably wouldn't because it's a leaf expression. but just as an example. Or you cross that with fasciation. So you try and pull out the gene expression that made this pathway happen with the duckfoot, and you get one hang of a weird looking plant. So that's some cool things that, and examples of how these mutants, this is Horled, W-H-O-R-L-E-D. There's a bloke, a popular fella in Australia through certain channels. Has this, and he calls this his trait. So this is what his trait is called, his expression. It's Horled, W-H-O-R-L-E-D. Same thing there. It's not a triploid. That's a Horled expression because you got more than 2 nodes coming out of the node. Usually you get 2. If you get 2, 3, 4, 5, it's a Horled expression. And yeah, polyploid is to do with, these aren't polyploids, these are gene expressions. Mutants. A mutant is when the DNA has been recoded, not its normal self. There you go, that's it. Let's see if there's what's going on. There's some comments. Oh, yep. Lars, yep, I've never seen anything about it. Oh, yeah, no worries. It's good fun. Hey, thanks for your props, mate. I appreciate that. I'm trying. I really am. It's hard in this, all these knockbacks and strikebacks and things like that. You know, we've really got to be careful. Lars says, oh, got to get your hands on some freak show. I bet it has good airflow. There you go. See? That's exactly why mutants are good in different applications. Good suggestion, Lars. I'm going to pull that up again, and we're going to look at it. Perfect. See, this is rad. That'll do. What do we want to look at? That's right, freak shows. Look at the airflow. So if you're in a region that's very, very hot, this would be perfect as. Or very dry, this would be perfect because it wouldn't transpire much because it's small surface area, but it's still going to get you the photosynthesis job to get you the end result. Mutants, see, Lars has pulled up on one reason why this mutant is a benefit. Excellent. We love cool people like that that can suggest rad things like all this weird stuff that we're doing. It's new. It's just new, but we can work it into advantages or disadvantages, and that's one rad advantage. Good on you, Lars. Onto stage. Lars says, what did he say? He was talking about alloploidy. I've never seen anything about that. Yes, that's right. It's always autoploids. It's very easy just to go and make a shift with chemicals, but to actually do it the other way, it's harder. I've done it both ways, and that's why I can say the autos is so much better. It's, allos is so much better. Autos is the same set of genes. Allos is so much better. That's through a triploid. So I went to the triploid, proved it was a triploid, and then crossed. And when you cross with a triploid, it's supposed to any of the offspring of it, because when it halves to its haploid, it's haploids, a triploid haploids into its 2 cells, so it's 2N. So if you breed with it, it will produce, no, it doesn't halve, sorry. It keeps its 3N. So when you breed with it, its offspring of a triploid is a polyploid or tetraploid with 4 sets. So that's what I had. That's what those seeds are, actually. I've got a few seeds of that, which is very rare. I haven't heard anybody else to have tetraploid alloploid seeds. Yes. Light would penetrate more too. Yeah, it would for those freak show cultivars. Kind of like Monsteria. Yeah, Monsteria's got that weird expression of variegation and ornamentals that cannabis now has. It's cool. So we can all have those and sell to a different market. Those people probably aren't even interested in medicine. They oppose everything that goes on with it, but they would like the actual plant to look pretty. It's really cool. I'll try aloes next. I got to look up how to do it. Well, that book, mate, get that book and then make your triploid, get your pollen, induce into a diploid, which will make your triploids, and then, yeah, continue. Aaron says, here's something I heard the other day. The WHO, World Health Organization, just announced that cannabis has been found to have medicinal and beneficial properties. So yes, that's the World Health Organization. They depict a lot of things. So if Australia's not listening to them, Australia's blind and will be ridiculed. But Australia still has to make money out of it first, remember? So they got 2 or 3 years to go. Yeah. And it'll be different. Here, that's why we've all got to prepare. If you can get in your little tiny cool things now and your knowledge up on this areas, there's so many beneficial areas in Australia that will need your help because it's lacking everywhere. GPs do a 4-hour course and they're qualified to prescribe it. As an example, they wouldn't even heard of a polyploid or what's this about a sativa versus indica, chemovar. You know, why does it give caffeine sensitive folk like myself a elevated heart rate? Oh, really? Never heard of that. Why? You know, all this sort of stuff. So all you educated folk, you know that already. But no, so you know more about the doctors in Australia regarding medical cannabis. So it's, if you can do some sort of study and focus on it a little bit, my goodness, you'll do well. Everyone will be after you soon. How long has this been going for? We'll talk a bit. Oh, what? An hour and a half. Really? Oh, wow. I love reading. And Newton's, or Ploidy. Ploidy's my focus area for the last few years. So it's been special to me. And it'll be my focus for quite a few years. It's rad. It's just, yeah, terrific. Is it? If you want some questions, pop your questions in now. Because, all right, been going for a bit. That was a good plant mutations and polyploid show. If folk don't understand about it, they could just go and podcast that and have a good understanding. So I'm pretty happy how that went. Fingers crossed, says Aaron. Yes, regarding all the medical laws and stuff like that. Let's hope that Australia, well, it is happening. Because you've seen all the studies in the news. Now they're doing all these studies with people. So once they approve it, that's what Australia politicians want. They want Australian studies, believe it or not, before they'll believe it. Yes, like people in the States and Canada do it wrong. It's retarded. But anyway, that's an excuse how they've said to prolong it so they can keep making all this money out of it. Because Big Pharma is responsible, but they're greedy. And they're paying a lot of, anyway, I don't want to get in that. Well, it was a great show. So put your questions now, if you got any. Next week, I'll give you a bit of a sneak preview. It's going to be a pretty good one too. I've been starting to put the slides together. It's this one. It's on pruning, side lighting, and Australia folk needed for 2 studies. That was this week. Right. Oh, but I'm putting together some slides for pruning, side lighting. I've got a few on that. There's a bud moisture test, drying. Yeah, so that's been going. Oh, some animal love. So yeah, that's up for next week. So I'd love to see and hear you around and hear your suggestions, because I reckon that's about it. Thank you again for everybody contributing in chat. I appreciate it. Because without you, it wouldn't really be a show. I don't like talking to myself. And thank you for listening. And for those who listen on podcasts as well, I appreciate you for trying there. It's been great. So keep up your good work, cool cannabis folk. Respect this medicinal plant that we have. Grow it in ways that brings out its medicinal properties so that we can really cast a shadow over street weed compared to medicinal weed. Massive difference. Even the companies in Australia, I'd consider it street weed they're selling, because they don't have terpene profiles or flavonoid profiles. So that means it's lost. So it wasn't dried or stored properly, and that's because of its age. So the powers that be in Australia don't know about that yet. They're just trying their best. So, yeah. Better education for all. Thanks for your nice words. Lars, Aaron, everybody else who's turned up today. It's really good. I'll hope to see you all next week. Happy breeding, happy growing, and good health to you all. Bye-bye.